Thank you all for coming. I promised Maddie I wouldn't get too emotional, so I'm going to try really hard not to cry. I am delighted to introduce Maddie Armstrong for her exit seminar. It's gonna be okay. Maddie was one of the first grad students to join my lab when I got here, and I felt a tremendous amount of pressure to choose people that would form a really amazing lab community, both scientifically and people who would support and nurture each other. And Maddie has been so much of both. One of the stories that really sticks with me when she first got here was how in her freshman year, her first day of evolution class, she marches up to the professor and says, "Can I join your lab?" And she really brings that enthusiasm and commitment to all the amazing work that she's done here. She brought new questions that I never would have dreamed of, and it's been really cool to see her develop those. I'm really excited about the postdoc, which she'll tell you a little bit more about. And beyond that, I think you can all testify to what an amazing community member Maddie's been. She's always the first to volunteer for everything to the point where at the end of last year, I'm like, Maddie, you have to stop. You're doing great. Everyone's so lucky to have you, and just please stop saying yes. And I think she's been really able to do some amazing work and leave a lasting impression on all of us. So here's Maddie.
Looks like it's being recorded. Yeah, we got no punch cards from the lab, mostly inspired by me not being able to say no. So every time I got to say no, we were 1/10 closer to a lab treat, and that was our trip motivator. So okay. So thank you all for coming to my talk. I'm really excited to share my dissertation work with all of you today. But something important, make sure you put your guess in of how many larvae you think me and collaborators, so like undergraduate mentees, counted and observed throughout my PhD, because there's a prize. I would say it's a pretty good prize. And I'll announce it at the end who's closest. Okay. So today I'm going to be talking about my dissertation work, basically titled Evolutionary Implications of Urbanization on a Coastal Marine Invertebrate. Okay, so let me make the screen small. So urban environments, highlighting here, present this really unique opportunity to observe rapid evolution in response to novel selective pressures. And there's been evidence of phenotypic, physiological, and a genomic variation associated with urban environments across several taxa. So here I'm highlighting kind of some classic examples with the peppered moths associated with industrial pollution and lichen die-off. We have anoles and acorn ants, and with thermal tolerance, with urban heat islands. And of course, Andrew's work in the killifish looking at pollution tolerance. But something I was really interested in, especially when I joined Rachel's lab, was the marine environment. And so I started getting interested in urban marine environments. So these coastal marine environments lie at this transition of sea to land and can face a lot of complex stressors, as you can see from this figure from a review, like a paper by Alter et al. In 2020. And so there's a lot of different things that these marine environments organisms are living near cities are facing that we might not be thinking about as like a kind of classic urban environment. So this is something I was excited about, and that really led to my overarching research question for my dissertation, which is, what are the evolutionary implications of urban stressors on a marine invertebrate species? You probably know what species, but I'll share why in a second. And then that really falls into two sub-questions. So here first is, how does the urban environment as a whole affect evolution in marine invertebrate populations? And in this project, I used a lot of genomic techniques to disentangle variation associated with urbanization. And then the second sub-question is more on a development angle. So I zoomed in and was really interested in looking at how an urban-associated pollutant affects larval development and gene expression. And to first do that, I looked at baseline exposure. And then secondly, taking what we learned from baseline exposure, how does coming from an urban environment or not affect your larval development and gene expression patterns? Okay, then I'll use these icons kind of throughout, where I have more of my genomics project, which is my chapter 1, and chapter 2 with that single beaker icon, and chapter 3, the beaker plus the city. So why purple urchins? You saw the treats made by my lab in the back that are all urchin themed. So urchins are a really great model system that I use throughout my work. They are, I think, the perfect system for the question. They're really widespread marine invertebrates across the Pacific coastline, and importantly in urban and non-urban waters. And they're pretty easy to find, especially for someone who didn't have a lot of marine tidal experience coming to grad school. So this is just a photo of iNaturalist showing all of the different like collection sites. And this is from a bit ago, so there's probably more. And there's a photo of me doing some of my first field work. So they're pretty easy to identify. They're really widespread, and they're in urban and non-urban waters. But besides that, they're also a really useful genomic and developmental model. So there's a lot of really useful like resources around the development, around the genomic work with them. And here I'm highlighting a little picture of some of the larvae, and then also how like it looks like when you spawn them. So that's why the cupcakes have a little orange inside, because that's what the eggs look like. And also they have pretty high gene flow. So there's been previous work on purple urchins looking at their like gene flow across their habitat. And there's, despite high gene flow, there's been some research on like local adaptation to pH. So I felt like this was a good model system to move forward and look at other kind of fine scale environmental variation. Okay, so let's first focus on my first chapter, which is really thinking about how does the urban environment as a whole affect evolution in marine invertebrate populations, specifically purple sea urchins. And again, remembering that there's a lot of different stressors that are happening in these urban areas that might not be present in non-urban areas. So to address this question, it kind of falls into two sub-questions. So first, it's really important for me to understand how do urchins even differ between urban and non-urban sites in the first place within a specific region. So here, I'm just showing like, I'll talk more about the sampling regime in a second, but basically we're looking at an urban site and a non-urban site that are from the same geographic location. And the reason it's important to compare within a similar geographic region is so that you're kind of controlling for other things like temperature and salinity, et cetera, that might differ if you're sampling in like very different geographic space. And here I'm highlighting, I think you can kind of tell which is the urban and non-urban, but a beautiful non-urban protected site and an urban site that's a little bit more broken down. Okay, so thinking about this difference between urban and non-urban sites, of course, there's multiple cities across the range, and we would expect that genetic variation is going to be more similar in one region, like close proximity compared to sites that are further apart. And then that kind of takes us to the second question of this project, which is identifying parallel signals of urban adaptation between urban and non-urban sites across multiple urban sites. And if we expect that there is shared variation between our urban sites and not the non-urban sites nearby, and strong selection, we might expect that these urban sites are going to have more shared variation with each other compared to the nearby non-urban sites. And this would suggest that there's local adaptation to marine urban environments. Okay, so what the project sampling kind of looked like is, to answer this question, we collected urchins in urban and non-urban areas with a lot of different helpers, including my partner, who luckily was available most holidays and would just come down with me, because low tides are always Thanksgiving and Christmas for some reason. And these sites were classified as urban and non-urban from previous literature, mostly by this Mussel Watch group, where they were able to look at like population density. But my cities are really Victoria, BC, Los Angeles, California, and San Diego, California. And our urban sites are much closer to urban wastewater, which is also another distinction for my urban and non-urban. And we collected tissue from 19 sites across all of these pairs. So I had 209 samples, and we had a mixture of urban and non-urban, of course, within each region. And then after that, did a lot of lab work, and we did whole genome sequencing at about 7.5x coverage. And after some slight filtering, just due to low coverage samples, we ended up with 183 individuals. And if you're very genomicsy and you're curious about my pipeline, happy to talk about that later. But mostly use this pipeline through SNP Archer that helped kind of standardize the pipeline for analyzing the data. So first, I'm going to show the neutral variation. So it's a thinned data set to reduce any like linkage bias of like about 19,000 SNPs, and then we'll get into more of the full data set. Okay, so the first takeaway from this study is that there's actually no population structure across the range, which is surprising. I definitely expected Canada to at least be a little bit different, but nothing. So I knew LA and San Diego, based on previous work, would be pretty similar. But what we're seeing here is our sample sites, and then within each region, all the smaller, like the exact places I sampled. And then on the far side is a structure plot where the light blue and the dark blue are not really, pretty much they're evenly mixed throughout. And so if there was structure, it would mean like the orange is all dark blue and the lower sections are all light blue. But there's no structure. And then another way of visualizing that is with a PCA. So this is just showing Color is non-urban and urban, so light blue is non-urban, brown is urban, and then our shapes are our 3 regions. It's just a blob. So there's no population structure within our data, which is interesting. So from here, I was really interested in looking at the full data set and seeing if there was any outliers associated with region or urbanization. Okay, so moving to our full data set of 219,000 SNPs, we actually start to see that there are outliers associated with urbanization. So there's actually no outliers associated with region. Again, kind of weird. But we do see that, so in these Manhattan plots, everything above those lines, we're seeing outliers, and each of these Manhattan plots is representing one of our regions, so Victoria, LA, and San Diego respectively. And we're seeing that there are outliers in our data set associated with urbanization. And this was done through FST outlier scans using the whole genome. And maybe you're curious what these stars are showing us. So while outliers are associated with urbanization, we see not a lot of shared outliers. So if there was a lot of shared outliers, we'd see a lot of these stars that are represented across all 3, but we're seeing a few shared between LA and San Diego and 1 shared between Victoria and LA. But it doesn't seem like there's like a specific urban SNP that's shared across all of our regions. And so this kind of got me thinking like, okay, well, maybe it's polygenic. And maybe it's not like one SNP is the right SNP for each urban environment. Okay. And then another analysis to really dig more into if there's variation with region or urbanization is an analysis done by my lab mate, Katie, which is really great. So here we looked at in kind of integrating multiple SNPs instead of just like single SNPs using a sliding window approach. And this can really help identify small effect size SNPs. So these Manhattan plots, the points are not necessarily SNPs, they're more windows. And what we're seeing above these lines are significant windows, SNP windows. And we actually see here that there are significant SNP windows associated with region, but there's also significant SNP windows associated with urbanization. And I want to draw your eye to the F statistic plot on the other side, where while there are outliers associated with region, we're actually seeing that the mean effect sizes were larger for urban. So that's kind of that brown bump further to the right. And so we're seeing, again, urbanization seems to be driving a large part of our data. And I'm not showing it here, but the non-significant SNP windows didn't show any pattern. So this is another like, okay, there's clearly something going on with our data set and urbanization. So I wanted to dig deeper into if there's polygenic effects. And yay, there were. So when I first saw this, I was really excited. So here we're looking at these different PCAs, and each dot is an individual, and it's grouping by non-urban in blue and urban in brown. And what we see here is that PC3, 5, and 6 are separating out by urbanization. And we actually, when we run this similar analysis with region, we see no separation. So it really seems like urbanization is driving a lot of the genomic variation in our data set. And maybe you're wondering like, okay, well, what if LA is just driving this pattern? It's not representative in all 3 of the regions. But it's actually all of the regions seem to be driving this variation. So what our box plots are showing us is just how these samples separate out on each axis. So I'm just showing PC5 and 6 here, but PC3 shows a similar pattern, where we're seeing that Victoria is not as strong in PC5, but in general, our urban and non-urban samples are separating across all 3 of our regions, which suggests this polygenic signal of urban adaptation kind of within each region specifically. Okay. So if you were not convinced, I have more data. So we also did a redundancy analysis. So this was done basically using all the different environmental variables within our data set, and then you compare down to see which variables are actually driving your data set. So interestingly, the only 2 that were driving are urban and depth, and latitude actually was not significant in explaining the variation present. And you might be wondering, I haven't talked about depth necessarily. So I meant to say earlier on my sampling page, the California Conservation Genomics Project did send me some subtitle samples, and I don't collect subtitly. And so we've actually seen this though in previous research. So the subtitle intertitle comparison was not a focus of my project, so I can't really have that be like a large takeaway of my work. But it's not surprising that they separate out by intertitle and subtitle samples, because I've seen this in previous work that I've published with collaborators. So really what this redundancy analysis is showing us is that along RDA2, our urban and non-urban sites are separating. And then along RDA1, we have our subtitle and intertitle samples separating. And there's no subtitle samples in Victoria because I collected all those myself. So that's kind of why the main focus of our data set is what we're really seeing is urbanization, but there is some signal of depth as well. Okay, and then our final thing to really make sure that this difference between urban and non-urban samples is not by chance is we ran this polygenic score test. So this was done to see if these polygenic signals of urban adaptation, again, were chance or if they were real. And so this was done basically by separating out our full urban urchin data set into a training and validation set. And so for a training, we had 60% of our samples, and each SNP was assigned this environmental genotype relationship using LFMM. And then we were able to create a beta coefficient with a weight explaining how much variation was present in our data set for those individual SNPs. And then using that, we were then able to create a polygenic score, which is the sum of this weighted genotype. And then we were able to apply that to the validation set and come up with how well those samples were actually, if they're actually separating by urban, non-urban, or if they're separating by chance. And we did this over 100 times. And we saw that 90 of our 100 runs, which is what the box plot is showing you on the right, is that they were significantly separating. And when we did this with just like random chance, like let's assign group 1, group 2, there was no separation. So again, this is really showing us that there's this clear polygenic signal of urban adaptation in our data set. Okay, lots of figures, but to summarize, first, we saw that there was no population structure despite this like huge geographic variation from Canada to San Diego. But we did start to see that there are urban outliers, but not necessarily like overlap specifically between those outliers. However, there's a lot of evidence that there's very clear polygenic signals of urban adaptation. And this makes sense because there's been previous work by Katie Lauderhouse and others where there's no one SNP that has like a perfect environmental climb. So it's likely that different SNPs are acting in similar ways that are explaining this variation that's existing on this like small spatiotemporal scale, where we're seeing urbanization as the main driver of the variation in our data set rather than region. And if you're curious about my color scheme, it was inspired by my cat, so I wanted to highlight her. This is the photo I used. And so far, everyone's liked the color scheme, so I'm like, good, because otherwise I'll have to make reviewers look at this and then they'll feel bad. Okay. All right, so back out to my overarching question. So reminder, and now we're going to think more about the specific, like a specific urban associated pollutant, and talk more about larval experiments that I've done at a Bodega Marine Lab. And remember, this is related to the poll question at the beginning, so hopefully you scanned the Google form, because there'll be a prize. Like I said, I'm excited about the prize. Okay. And so I showed you this figure before. There's a lot of different environmental stressors that are happening in these coastal marine environments. But of course, like maybe I could go out and like scoop up urban wastewater, but that feels like a lot of work. So basically what I did is I really focused on sewage and wastewater for this part of my dissertation. And there's one specific class of chemicals that's really common in urban wastewater. And this is a study by Muscle Watch, a couple papers that I cited below, where they saw that alkophenols are really common in urban wastewater. And specifically, you can see that there's just this higher prevalence where the blue is by Long Beach and San Diego, and we have less in those intermittent areas. And not only are alkophenols in higher concentrations in urban areas, but they've also been shown to have negative developmental effects. And they specifically have negative developmental effects in like a lot of marine invertebrate larvae, and those larvae can be really sensitive to these ecotox studies, so they're really useful for that type of work. And here I'm showing mussels and oysters, and then a couple different echinoderm species, including sand dollars and sea urchins. However, there's not a lot of work, while there is a lot of work in the larvae, there's not a lot of work pairing larvae with transcriptomics, and there's not a lot of work looking at how tolerance or susceptibility to these urban-associated pollutants can really vary among populations with differences in exposure. But we do know that obviously other environmental things vary, and we know that other stressors can have geographic variation. And it's important to understand how historical exposure via acclimation or adaptation can alter responses to stress. And just showing you an example here with temperature stress, this really cool study by Gleason and Burton in 2016. They took snails from cooler habitats and warmer habitats and brought them into the lab and exposed them to temperature. And not only did they have different gene expression patterns at the start, but they also had different gene expression responses with that temperature stress. So clearly historical exposure matters, and it's important to consider that in this type of work. Okay, So first, before I can even get into that, I really needed to understand how baseline exposure impacted our larvae in developmentally and via gene expression patterns. So I used the most common alkylphenol and wastewater, nonylphenol, and looked at its impact on development, survival, and gene expression. Okay. So this is kind of what our setup looked like. This was done in collaboration with one of my previous mentees who also did this for part of her senior thesis, Cindy Bala, who's now a master's student in New Zealand. And that's her over there working hard. You'll get to see that picture zoomed up in a second. But basically what this study looked like is we took one population of sea urchins from near Bodega Bay, called Stillwater Cove, and we created 4 mate pairs. So this was like a male and a female shown at kind of the top. And this male and female pair had eggs, fertilized eggs put into each of these treatments. So we had control, we had a low concentration, a medium concentration, and a high concentration of nonylphenol. They were all ecologically relevant, like low concentrations as well. And after we did that, we had 4 of those. And then we also collected a ton of data over a 7-day period for a variety of developmental stages. So we have fertilization success and development success, and I'm showing images of what those stages look like. And then also morphological data, survival data at the blastula stage, the gastrula stage, and the pluteus stage. And there's also RNA data for the gastrula and pluteus as well. And I don't have a ton of time, so I'm not going to go super into depth with this study. So I'm just going to highlight the results of what we found and then talk more about the historical exposure study and what we learned from this. But basically, we saw that there was no impacts on fertilization or early development, but we did start to see effects after that. So we saw that there was reduced survival actually at the lowest concentration, which was only 100 parts per billion of nonylphenol. And we saw this across mate pairs. So across these different families, there was clear responses of survival. We also saw that gene expression patterns were driven by both mate pair and treatment. So there was a lot of variation depending on what parents you came from. And then honestly, one of the big takeaways and something I didn't expect from this study is there was a lot of variation within one population, and it was much more than I expected. And then if you want to know more, it was recently published, yay. So you can also ask me after too. Okay. So given what we learned, now I was really interested in understanding how historical exposure, aka coming from an urban environment, impacts your development and gene expression. Okay. And so for this study, the setup is a little bit similar, but it's really scaled up. So here we are looking back at some urban and non-urban sites that you might look familiar from my first chapter, but we did 2 urban sites and 2 non-urban sites from the Long Beach area. And at each site, we actually collected 40 urchins. So 40 urchins were collected at both urban sites and obviously both non-urban sites as well, and then brought up to the Bodega Marine Lab. And here, the reason we did this was to really make sure we had a lot of genetic variation within each population, and we could have a lot more families. So the experiments kind of looked like this, where we had a male and a female, and we had a control or that low dose, which is the low concentration that we saw impacts in the first study that I talked about. And on average, there was about 12 families per population. And this was done, so we had like a total of 96 beakers across the whole study. And maybe you guys are curious about this setup here, highlighting Tracy, my valuable assistant. But basically, this is how we maintained these cultures, and I'll talk about like what the experiment looked like. But we have these, like Eric had this really nice previous setup that I then expanded across the entire like cold room area, where we had these like PVC frames that actually stirred the beakers and kept the larvae happy. And then we had, of course, a map of where each beaker went, so nothing got messed up. And this was a way to really keep the cultures happy throughout the experiment, which was a lot of like a couple winters back, like multiple months of big experiments. And then across all of that, we collected similar data. So we collected data on development success, and then kind of focused on these 3 main developmental stages, where in the blastula stage, only 17 hours post-fertilization, we collected morphology data, abnormality data, survival data, and RNA. Gastrula, we took similar data, but slightly different, obviously, morphological measurements. And then for the pluteus, there was much more morphological measurements, and then obviously the tail end of the survival data. And I'll talk about the survival and how that was measured in a second. Okay. And of course, this was a lot of work, so I had a lot of helpers, and specifically I wanted to highlight the folks that were really involved in a lot of the process. So Juliana and Fran DeCastro were really involved in like the experiment development and the data collection and kind of the day-to-day of like sitting with me while we're all in like like gear, making sure we're not contaminating anything. And then Evan, Cheyenne, and Lainey helped a lot with the photo analysis and measuring things, which is why they're banned from competing in the larvae count poll. But yeah, this was a huge undertaking, and they were really, really instrumental for making sure all of this could actually happen. Okay. So for the survival data, which I'll talk about first, because again, I'll go back for a second. There was a lot of stuff going on, and I can't talk about everything. So I'm going to highlight a few methods and then a few results of the main takeaway of the study. So everyone giggled because they saw the Stardew Valley. But so this is what survival counts kind of look like. This is at the density change over time, and if that was due to treatment or due to coming from an urban environment or not. And then these tubes here on ice was just like a small sample of each beaker, so I didn't have to disrupt the whole experiment. And then I did listen to a lot of Stardew Valley music. It's a really great game if you haven't played it. One of my friends convinced me to get an external number pad because I was like very nervously touching my computer with contaminated gloves. So this was really kind of what this looked like. And then for our first graph, so what we saw is that larval survival was slightly impacted by treatment. But in this case, we didn't actually see an effect of an urban or non-urban environment. So to kind of orient y'all to the graph, what we have is proportion survival, and then we have non-urban and urban. In color is the control is green and the treatment is purple. And what we're seeing here is that there's not really a difference between non-urban and urban. But what we are seeing is that survival tended to treatment tended to reduce survival. And this could just be due to treatment being stressful and survival is decreasing because of that. So that's kind of our main takeaway for survival. Now getting into the morphology data. So this is kind of what that setup looked like. There's a really great microscope out of Bodega Marine Lab with a camera mount where you can use like a full fancy setup. And so we just showing that here with the scope camera to take photos, a beautiful Pluteus larvae on the screen. And then we use slides with raised cover slips to not squish them and get really good photos. And then here's Cindy working hard, taking photos. There's no photos of me doing this because when I did this, I was by myself. And this is when I first got really into audiobooks. So it was definitely a big undertaking. Okay, so getting into first the abnormality data, and then I'll talk about the morphology data. But for the abnormality data, we're again seeing a really strong effect of treatment, not necessarily a huge effect of urban or non-urban environment. But to orient again, we just have non-urban and urban on the X and proportion abnormalities on the Y. And in the gastrula stage, we see a strong effect of treatment. And we also see increased variance in abnormalities in our urban populations relative to our non-urban. But then for our Pluteus, we see just a strong effect of treatment. And if you're curious, this is kind of what abnormalities tended to look like. And a lot of these are classified from previous work, but then these are just my own photos that we like Photoshopped to make them look a little nicer on the screen. And then for the blastula stage, we actually see a slightly different trend, which was a little surprising at first. So there's actually less abnormalities in treatment for our urban blastula. So we see a significant effect of the interaction of urbanization and treatment. And we also see a significant difference in the variance between non-urban and urban. So it seems like the blastula stage is a slightly different trend relative to these two later stages. But then after we kind of like talked about it, I think this does make sense where blastula are only 17 hours post-fertilization, and they might still be like protected from their maternal like transcriptome or something like that, because they only recently start expressing their own transcriptome in this stage. And so since they're so early, maybe they're a little bit more shielded from their treatment conditions, while later on the gastrula, which is 48 hours, and the Pluteus, which is 7 days, are starting to show like a much clearer effect of treatment. Okay. So that's kind of what we learned for abnormalities. We see this high proportion in treatment, but maybe slightly different effects of the different stages. But then for the morphology data, we actually see that the urban larvae are larger across stages and larger in treatment. So we're seeing kind of a much clearer effect of urbanization for our size data. So I'm just showing our gastrula and Pluteus here. But for gastrula, we see an effect of treatment, urban, and the interaction of treatment in urban. And the same for the Pluteus. And this is a huge sample size for both of these. And for the blastula, it was a little bit different. That's why there's an asterisk. So they were larger if they were from urban environments, but blastula were slightly smaller in treatment. And again, I think that kind of makes sense that they were a little weird, like they were in the abnormality data. because there's a little bit of that shielding from being an early life stage. Yeah, so we're starting to see that, okay, coming from an urban environment, you're both larger and you're larger in treatment. This could be because the urban individuals are less stressed out, or maybe they're growing big to capture more food and they get out of there. So if you're a large gastrula, you might be further through the gastrulation process and they don't eat until they're a pluteus stage. So they might be trying to develop really quickly to get out and start feeding and escape potentially. So a couple different things of why we think that urban might be larger in treatment and in general. Okay, and then the last kind of data that I'll talk about for this study is our RNA data. So what this kind of looks like here, the whole point of this whole slide is pretty much to go from 2 50 ml tubes to 1 2 ml tube to concentrate our larvae to 2,000 individuals in 1 2 ml tube. This hand centrifuge was instrumental for that because basically the reason that they have to go through this whole process is that the larvae are at about 20 per mil to be a happy like culture pretty much. If they're in too high densities, they'll die. But for RNA, you need a bunch of individuals. So Fran was really helpful in this, is just transferring into smaller and smaller tubes. And then also speaking of the RNA collections in the hand centrifuge, I wanted to highlight Juliana. This was initial, like very early RNA chaos, and this is before there was a handle. So you'll see in a second that there's actually no handle on the end. She was just like holding this tiny little nub. And that's because I got hit by it once and I was like, you have to do this. I need to experiment. So thank you for that. And then yes, a lot of people have also told me that they make mechanical versions, but I was like, I know we need this to be like, we have to have it quickly. Otherwise it's like they swim back up. So yeah, it's very helpful. Okay, so now let's talk about kind of my two main takeaways for my gene expression data. I'm glad you guys like that. Okay, so first we see that gene expression patterns show upregulation with treatment. So this, we're looking at a heat map. And if you're unfamiliar with this setup, basically the columns are individuals and the rows are genes. And then the colors are denoting, like I put urban and non-urban. So the grays, browns are urban, the blues are non-urban. And that column below is treatment. So we have control treatment for each population. And we're looking at the gastrula stage here. I didn't think you want to look at like 100 heat maps. So here we have 506 outliers, and we're looking at outliers associated with, or we're looking at genes associated with treatment. And we see these like yellow stripes of upregulation in some of our treatment. But I think another thing that I see when I look at this is there's a lot of variation between individuals. So then, of course, we wanted to look more deeply. So here, I used a weighted gene correlation network analysis, or WGCNA, to identify co-expression modules. And this was done basically to see which genes were varying with treatment together. And we see that they actually also vary with urbanization. So this framework is looking at co-expressed genes. And first looking at the gastrula stage, we're looking at 75 of the 506 differentially expressed genes, and they all fall in like one module together. And we see that there's this variation also with urbanization. And these genes tended to be associated with amide transport and also response to chemical, which is good. And then for the pluteus stage, we see it also is associated with urbanization. So this module contained 1,000 of the genes from 3,000 total. And these genes were typically associated with catabolic processes. So in general, we see that these differentially expressed genes with treatment that are really associated together are also associated with urbanization as a whole. Okay, so to wrap up this study and summarize these findings, we see that larval survival decreases with exposure, although slightly. And then we also see that there's higher abnormalities in treatment, but maybe some differentiation between stages, with our urban blastula less impacted. We also see that urban larvae were larger in general and in treatment, with an asterisk of blastula being a little bit strange. And then we also see that gene expression varies by treatment and urbanization. Okay, so to wrap up everything, again, my overarching question for my dissertation is, what are the evolutionary implications of urban stressors on marine invertebrate species? And going in a reverse order, we think that with this urban pollutant treatment, we saw that it decreased survival and affected gene expression and development. And then thinking about the historical exposure context, we saw that our urban urchins continuously exposed to these stressors have different morphology, and there was differentiation in gene expression when they were exposed in the lab. And then zooming out all the way back to my first chapter, we see that selection leads to outlier SNPs in urban populations. And there's these very clear polygenic signals of urban adaptation despite hygiene flow. And you might be wondering what I'm doing next is I am actually starting a postdoc in January, yay, with Katie Lauterhaus at Northeastern, continuing my genomic work. Now we'll be moving more towards some computational focus, doing genomic forecasting with oysters. So still sticking with marine invertebrates. And then I have a thank you slide. So I wanted to say thank you to so many people. I just did a big old collage also for my like little research equipment things that were also instrumental. My QE committee, Rick Grossberg, Eric Sanford, Jeff Rasibara, Daniel Ronsi, and Andrew Whitehead. And of course, my dissertation committee, Rachel, Eric, and Andrew. All of my urchins, both larvae and adults who sacrificed their lives for my work. The Bay Lab and all of my undergraduate mentees, and the population biology graduate group, as well as my cohort. The Bodega Marine Lab community has also been really supportive and helpful, and I've really enjoyed my time out there, and the Aquatic Resource Group for always doing everything they can to keep my things alive. Even on Christmas, when there was chaos and they were out there making sure things didn't die. And of course, my fieldwork helpers and larval experiment helpers, and all our weird tools. And Gary Sher, Carol Vines, David Gold, and Eric Sanford for providing me space out at BML. Finally, also my partner Ben and my loving support system. Thank you guys all for being here. And then a few other folks, including Margaret Seeley, my urchin dye collaborator. We did a workshop at Bodega, which this is a little like piece of urchin dyed fabric. It's pink, which is kind of surprising. And of course, my tattoo artist for being willing to tattoo so many biology things. My softball teams, Bodega Bastars and Sac Hart, and my funding sources. And then before we do questions, I want to see who the winner of our prize is.
Last
So
chance.
I submit your answers. So I'll vamp for a second. So chapter 1, no larvae. So starting at 0. Chapter 2, we collected 9,911 individuals. So you guess below that. You're not close. My final chapter included 43,777 individuals. So our total is 53,688. And who? Nikki.
What is the
I made a print actually using the design my tattoo artist did of my urchins. So, and I'll take any questions. Thank you guys.
So I had an interpretation that jumped in my head when you were showing your survival and morphology data. And I suppose I'm just wondering whether you've already thought of this and know why I'm wrong, or if it is something you've thought of, which is that you had the treatment affecting survival, and then you had larger individuals. And it could just be that larger individuals survived, right? They were the ones who were likely to survive, which would then mean there's selection for larger body size, which is why the urban populations are larger.
I mean,
Is that
essentially, yeah. I was like, I mean, so this, yeah, that's a good thought. So I guess so the survival data and the morphology data are different samplings. So I can't, like I meant to say before, all of the morphology data was collected via preserved individuals. So I can't go back and like know the size of the individuals that were counted for survival. But yeah, that's totally possible for an interpretation, is that they are larger. Maybe they're like capturing more food and able to sustain their populations in urban habitats that are stressful. Yeah, thank you.
In your first chapter, first of all, awesome talk. I love the color choices. Fantastic. The images taking you along, these little purple urchin images.
Oh yeah, I've been crediting BioRender, I promise. But BioRender, because they're a great model system, has beautiful urchin icons. So thank you, BioRender. Because I do know, yeah, the fact that a few systems are represented, it's usually like more model systems. So yes, I do like this.
And yeah, I do have a science question. In your first chapter, it looked like the difference between urban and non-urban in Victoria was lesser overall in some of the figures. First of all, is that true across most of the ways you looked at the data? And then if so, do you have a thought as to why? Is it potentially like a temperature interaction, or is it that that urban site is maybe less urban than the Southern California?
Yeah.
Yeah.
No, that's a good, let me pop back to that actually. So, I think you're talking about the box plots with these PCAs.
Yes.
So
Yes.
we only see Victoria is maybe not as different in PC5, but PC6, there is like a starker response. I honestly am wondering if it's I mean, San Diego also has only a few samples. There's only four sites and like 40 individuals total for Victoria, but it could be that there's other environmental variants that are driving it. And that kind of makes sense. There's been like a lot of really cool urbanization research on like cities are not perfect like replicas of one another. So like here I'm talking about like parallel signals of polygenic adaptation, but of course like each city is unique in their like history of urban outflow and things like that. And for Victoria, I didn't say this, but they only like recently got a treatment plant up there. And like one of my field sites was even like nicknamed by locals as Garbage Point. And I was like, yes. But yeah, so it could be that there's like some other variation that's maybe impacting this PC5. But for PC6, we're still like seeing that stark response. Yeah, good question. Yeah, Andrew.
So in your chapter 3, did you find any gene expression modules that showed a population dependent dose response? When you've got population response, you got dose response, but if you've got variable sensitivity between populations, then you might predict there would be genes that show an interaction.
Yeah.
Response between the two.
I did look at gene expression patterns with like so I was mostly looking at the interaction of urban and treatment, but I also
Yeah.
was doing some, but yeah, so like.
No, that's what I mean.
Okay, yeah, so when I did interaction, like keep like differential gene expression for the interaction, there honestly wasn't a ton that popped up. It really seemed like a lot of what was driving that variation was treatment early on, and that's kind of why I pushed towards doing WGCNA to like see if those genes associated with treatment were also then associated with one another and also varying with urbanization. But yeah. Yeah.
Yay.
That was so good.
I have an ecology question.
Okay.
How, when you're talking about urban and non-urban sites, like how far do you think the effects would go sort of spatially? And like, do you think it's like more of a gradient maybe actually in the effects, or is it like a threshold sort of thing? Or it's like if you could sample a bigger spatial scale, I guess.
Yeah, no, that's a good question. I'll pan back to speaking with the little picture. So I think for my urban and non-urban sites, all my urban sites were like significantly closer to wastewater outflow, which is what I was interested in. So they're like, I have it all in like scientific notation, like kilometers. So like 4 kilometers or so, like all my urban sites were that proximity, while the non-urban sites were like usually like 15 to 20 kilometers further. I think a gradient would be cool. At least in LA, there's kind of a spot where urchins just aren't. And so I couldn't do like a continuous sampling. It was like this bay sandy habitat and like urban Long Beach, and you could go north and then south. It was just like there was a gap. I bet that there probably is like variation, like gradient in the wastewater outflow. So it's not as perfectly categorical as I've denoted it to be. But yeah, and I think you have another piece to that, sorry.
I just read.
Okay, that's fine. So yes, I do think that there could be variation in like an urbanization gradient. And I do think that would be cool to like continue to look in, is like now seeing that there is variation in urban and non-urban samples across these 3 regions. Like I would love to sample even further or more sites to see if like this is a continuous pattern, and also like seeing if like city history kind of varies that like pollution exposure effect. Yeah, Eric.
In the first chapter, I thought it was interesting that you had outliers associated with urbanization, but they were different outliers between the regions. Can you say a bit more about how you interpret the fact that they're not shared?
Yeah, so I know at first, like we were like, wow, there's like so many outliers associated with urbanization, and it was a little bit like confusing why there weren't as many shared. Like I expected like a beautiful figure of like one big peak that all of them share. I was like, that would be cool. But I really think that comes down to like, there's no like perfect SNP maybe for this like urbanization environmental gradient. And like other work has looked at like different stressors like temperature and pH, where they see that there's like a lot of SNPs that might be like controlling this, or a lot of SNPs that might be contributing to the same kind of like adaptation or acclimation variation. So that's kind of why there aren't these peaks where the trait is much more polygenic and there's a lot of SNPs contributing like a similar way. So it doesn't necessarily have to be the same SNP. It can be like different ones in each environment, but they're kind of, the outcome is very similar.
Can it also be possible that there are different stressors in the different regions too?
Yeah, so I think that you're right, like we can't fully disentangle if it's slightly different types of urbanization that could be driving this like differences. But the fact that there is like outliers associated with urbanization in each region at least shows that in that region there's separation of those. Yeah, it's a good point. Thank you guys.